So for the World Congress, I had been asked to give an overview of SDS in the United States. It was a whole session on SDS. I actually don't remember who may have talked this morning, but I'm going to go backwards and give you an overview of SDS, its importance, the symptoms, the life cycle, and then towards the end, I'm going to focus a bit more on a research project that I'm currently working on with several colleagues here on campus on cropping system diversification and their impacts on SDS. All right. So if most of you are from around here, you probably have come across SDS at some point. Maybe driving in some wet years like 2010 or 2014. If you were driving through the roads, in the country roads in Iowa, you may have come across fields that look kind of like this. There's some patches of drying plants, defoliating plants. And if you were to get closer to these patches, you would likely have seen symptoms of SDS. And the foliar symptoms start off as these chlorotic lesions between the veins of the leaves, and as the symptoms progress, they become more like streaks in between the veins, and they become necrotic too. So we call this symptom in the middle here, we call that intervenal necrosis and chlorosis. As the disease becomes more severe, eventually these leaves will completely dry, they will drop, and so you have premature defoliation, which is one of the main causes for yield loss on SDS. If this happens early on when the pods are still setting seed or the seeds are still filling, you can have much more significant impacts of SDS. So these are the most obvious visible symptoms of SDS. But there are symptoms also below ground. The fungus that causes SDS infects through the roots, and as such causes a root rot that is apparent by poor root development. There's poor lateral root growth. The roots can be small. And if you were to cut through the root, the taproot, you would see in the infected plants this brown, streaky discoloration of the cortical tissue. So that's a sign of infection compared to a healthy root that has a very white cortical tissue. The other thing you might see is if the soil is wet and you pulled up a root, you might see these blue masses shown by the arrow, and those are conidia of this fungus. So the fungus is sporulating on the root surface. So these might be visible. So SDS has been in the United States for quite a while, since 1971. It started in Arkansas. That's where it was first detected. And currently it's found in all soybean-producing states in the United States, in North America, in the United States, and one state in North America. And so this is a progression of how the disease spread over the decades. So this is the 1980s. It had spread to adjacent states, adjacent to Arkansas. In the 90s, it spread even further north and to the east and to the west, and Iowa. And then by the year 2000, it had even now gone to Michigan and Minnesota. It was up in Ontario. And even now in 2010, it has further spread to Louisiana and up to South Dakota. So it's on the move. It continues to be spreading. There's some ideas that it may be moving with the soybean cyst nematode. The fungus has been isolated from cysts. But in any case, it follows a similar pattern, spreading with soil movement, most likely. So very widespread disease impacting soybeans. In terms of yield impact, the estimated yield losses for the period of 2006-2014 range anything from 0.6 to about 2 million metric tons of soybeans, representing $200 to up to $750 million in losses nationwide. And these are estimates. Dan Rather and Carol Bradley basically contact different states and ask, can you estimate the yield loss that you get? And so these are estimates, but here you see a field with very severe defoliation, premature defoliation that can cause that yield loss. The yield loss depends very, very much on the environmental conditions. So when you have a year that's very wet, like 2010 was in Iowa, and actually 2014 also, that's when you see these peaks of yield loss due to SDS. So each year to year is going to vary quite a bit. Again, this is averaged or total over 28 states. So in Iowa, there are particular years you might not have seen much yield loss at all if it was a dry year for us. Like this year, we probably didn't have any impact of SDS. Yeah, so it varies a lot with the weather. And how does this compare with other diseases? Soybeans, of course, are affected by many diseases. And this is an example from 2014 based on that same data. And you can see SDS is here second together with the seedling diseases. And this is pretty typical of other years. Generally, SDS is considered to be the second or third most yield-robbing disease in the United States. The first one being soybean cyst nematode. And there are several others that can become, depending on the weather, more important. But usually we're up there in the second or third most important in terms of yield impact. All right, so what causes SDS? In North America, there's a single species that causes SDS. It's called Fusarium virguliforme. But in South America, there's actually this species and another 3, maybe 4 other species that cause SDS. So Argentina, Brazil, maybe a couple of other countries in South America also have SDS. This is a fungus, like most of the Fusarium, that produces macroconidia that are canoe-shaped, banana-shaped. They also produce chlamydospores, the round spores, that are really important for survival. These spores are thick-walled, they're pigmented, and they can live for decades in the soil. So this is a very difficult pathogen to get rid of once it's established in the field. And the culture you see is a bluish culture, again, because of those blue, the conidia have these, form this blue color in mass. Survival in cysts, I already mentioned, movement through soil, so spread through movement of soil. So this is our organism here. And I'm going to go over the disease cycle. So we have these spores, this inoculum in the soil, usually on plant residue, so the decomposing roots from the previous season or from 2 years before. In warmer parts of the country, maybe some conidia may survive, some hyphae, but the inoculum is there in the soil. When you plant soybeans, they can get infected very early on. So we have been able to detect, or others have been able to detect also, within 2 or 3 weeks after planting, or a seedling that's 2 or 3 weeks old, should I say, they may already have infections by SDS from this pathogen. The plants usually don't show foliar symptoms at this point. They will continue to develop even though the fungus is in the roots. And later in the summer, sometime in August, mid-August, late August, you may start seeing then the foliar symptoms appearing. And these are caused by toxins that I will mention here in a couple of slides. So the foliar symptoms appear. You can have then premature defoliation that causes the yield loss. And eventually you harvest, and the residues that stay in the ground then continue to contribute to buildup of inoculum in soil. So let's think about the root infection. What is known about that? It can occur in young seedlings, like I mentioned. The penetration, the fungus tends to use areas that are more vulnerable. For example, the root cap, the root hairs, and also emerging lateral roots. So there's natural wounds or as young tissues that are more susceptible. The fungus establishes itself in the roots. But to get the toxin from the roots to the foliar symptoms, the fungus needs to make it to the vascular tissue. So if the plants have only a cortical infection and the fungus is not found in the vascular tissue, in the xylem, then the toxins cannot move up. So this early infection establishment in those root tissues is really important for symptom development later on. And then the foliar symptoms then are caused by toxins. A lot of the work that's been done on the most well-known toxin, this toxin called FeTox1, was done in Madame Bhattacharya's lab. And so he's characterized this protein. It's a 13.5 kilodalton peptide that interferes with the rubisco and increases levels of the reactive oxygen species, and so it leads to those typical symptoms on the leaves. And what you're seeing here in the picture are seedlings that have been immersed in a culture filtrate of the fungus. So there's no cells there, no spores, and it's purely, this is the way that certain groups, Hartman and Madan, established that it was really the toxins from the fungus that move up to the plant and then cause these foliar symptoms. It's possible that there are other compounds interacting and contributing to symptoms, but really there's not a lot of information about that. Okay, so another really important component of the disease cycle is then the impact of the environment, and SDS is highly dependent on the environment. Soil moisture, we think, is the most important environmental factor that affects SDS development. Very high soil moisture favors SDS. It's been well established both in greenhouse experiments and from field observations and field studies way back from the 90s. And this table here is more local for Ames, Iowa, actually. And there was a study that I did with Darren Mueller and Allison Robertson and XB. And we were interested to compare epidemic years and non-epidemic years and see if there was sort of a weather trend for SDS epidemics. And so what you're seeing is in the blue bars, in the light blue bars, were years where we had high incidence of SDS. So 2010 was our worst epidemic ever in the state. 2014 and 15 also we had pretty high SDS. The other years were years that we didn't have severe SDS. And we're comparing precipitation for these different summer months to the mean of the 30-year average for Ames. We also compared other locations, but I'm just showing here for Ames. And what you can really see is that in the years we had the epidemics, if you look at June and July, you have more than double than the average precipitation, the 30-year average precipitation, especially those 2 months, while the years that didn't have a lot of SDS usually were below average precipitation. So it's really a very important driving factor for SDS. Temperature also is important for SDS. There's been growth chamber and studies. Again, XP Yang did that here at Iowa State. And he characterized the optimum conditions, optimum temperature for root infection and for foliar symptoms. And it's interesting that they are different. So for the top graph here for foliar disease severity, the optimum for foliar symptoms is 25 degrees. But for root infection, the cooler temperatures tend to favor infection. So it's also true in the field that we think that if you plant into cool soils, you're more likely to favor infection. The roots are going slower, more vulnerable tissues stay there for a longer period of time. When we compare those epidemic years in that study I was mentioning earlier, we also saw generally SDS epidemic years were cooler, but it wasn't such a significant effect as the moisture. So we think really moisture trumps kind of the temperature effects. Another really important environmental interaction that happens is with the soybean cyst nematode. As you know, soybean cyst is mostly in every field in soybean producing regions, and it's also well known that when soybeans are infected by the cyst nematode, you tend to see more SDS, more severe SDS. And the graph you're seeing is data from one of my previous students where he infected plants with both pathogens, Fusarium verrucoliforme and SCN, or just Fusarium, and you can see a very significant increase, at least in normal watering, in SDS when both pathogens are present. Same trend in reduced watering, not statistically different, but it's just basically validated what has already been known. So management of SCN becomes important too when you're thinking of managing SDS. All right, so talking of management, what are tools we have available for managing SDS? Really the primary way, the most important way is using resistant cultivars, and Sylvia knows all about that. She works very hard to get us some good cultivars. And there are good cultivars available. Over the years, we've had improved resistance. However, resistance is not complete. There's no cultivar that is immune to SDS. So under optimum conditions, if it's really favorable, even those cultivars will develop disease, and you can suffer yield losses. One of the challenges is that there are many QTL, many genes involved in resistance. Each of them plays not a very huge role. We don't really know how much role each of them plays. And so challenge for breeders like Sylvia is how many QTL do we have to put together in a variety, which ones we select, and so it's a continuing challenge. The other management option that has more recently become available is the use of a seed treatment. And Elevo is a product from Bayer. It's a flupiram, and it's the one product available in the market that suppresses SDS. Again, it's not 100% effective. You do see protection from yield. You do some reduction of SDS. But it's the one product, and as you know, we probably have some concerns about using this single product. How long will it last? Is the fungus going to develop resistance eventually? So for now, these are the two most important approaches. And also the seed treatment is expensive. There's a couple of cultural things we can do. Improving soil drainage always helps because we want to keep those soils well drained to reduce infection. Avoiding planting in cool, wet soils also can help, but growers are limited in when they can plant. If they're ready to plant, and they just have to do it sometimes. So it's a little challenging for growers there. And then there's also a common misconception among growers usually that crop rotation with corn, which is what we usually do here, that helps manage SDS. Of course, crop rotations are well known for helping manage soil-borne diseases, but in this case, the rotation with corn does not help. I've talked to growers who have planted corn for 10 years thinking they'd get rid of the pathogen, and they planted soybean again and got hit by SDS again. And there's also more recent work from Navi and Yang's lab here on campus, where they have actually proven that when you have corn residue, especially kernels and corn roots, in the ground, the fungus thrives on that. It loves feeding on that and actually increases population. So corn is really not helping. Which brings me to the last part of my talk, the project I was mentioning, which is really about cropping diversification. And what can we learn from this field trial that Matt Liebman, who's an agronomist here, has been working on for a couple of decades now. I think 2001 is when he established this. And Matt's goal is to compare 3 different cropping systems. Each of those systems is characterized by a crop rotation. So we have a 2-year corn-soybean rotation, a 3-year corn-soybean with oats and clover, and a 4-year rotation with corn, soybean, oats, intercrop with alfalfa, and then alfalfa in the 4th year. So there's 3 crop rotations, and then also the management of the fertility is different. So in the 2-year rotation, they apply synthetic fertilizers, typical of what growers do. In the 2 longer rotations, they apply manure, composted manure, and they supplement with synthetic fertilizers as needed. So they're whole systems, not just the individual factors there. And so in 2010, when we had that very big epidemic of SDS, Matt called me and a colleague to the field and said, look, I'm seeing these big differences in disease. These plots were very defoliated, very high incidence of this disease. The plot right beside it looked pretty healthy, very green. Now, can you guess which of the plots was in the corn-soybean rotation or in a long rotation? Okay, everybody's pointing. You are correct. So corn-soybean rotations had very severe SDS, and the other rotations, adjacent plots across all the blocks in this field, had much less SDS. So we started collecting data over several years, and this is what the 2010 data looked like. So the 2 bars, the red and blue, are just 2 different varieties. One, of course, is a lot more susceptible than the other. But you can clearly see that both for SDS incidence, so the percentage of plants that had disease, and for severity, so it's the percentage of the canopy, the leaf area that had symptoms, very, very significant differences. Nearly 100% incidence here on the 2-year rotations and less than 10 in the other plots, and also for severity, very significant impacts. So this was pretty exciting for us. And when we looked at yield, the impact on yield was also very, very high. In the susceptible variety, 30 bushel per acre difference between the 2-year rotation and the longer rotations. And when we did regression analysis on this data, we saw that for this year, 87% of the reduction in yield was explained by SDS incidence, and there was about 45% explained by severity. But mostly this yield loss was caused by SDS. So we continued to collect data over the years, and this is a summary of the 6 years of data that we have. And this is disease index, so it's a combination of severity and severity and incidence, and combined for the 2 varieties. But again, we see very similar trends every year. They are significantly different here and here. These 2 years were very dry, so we saw a little bit of SDS, but not enough to really measure differences. But consistently then over the years, we had the same kind of effect. So we became very interested to find out what explains this. What are the factors that are contributing to this suppression of SDS? We did a couple of other measurements. I started working with Gwen Beatty and Larry Halverson. And Gwen, using community profiling data and qPCR with specific primers, we looked at soil and see is there a difference in pathogen levels in the soil. And as you can see, if you look at this graph here, between the 2 and the 4-year rotations, you definitely see that there's a significantly higher amount of the pathogen, so more pathogen buildup in the 2-year plots than the 4-year. And you can actually see those differences in the corn plots too, because every stage of the rotation is present every year. So she also sampled soil from the corn plots, and even in the corn plots, that difference is apparent. So there are differences in pathogen level. And also, she did some initial work in 2002 comparing microbial communities. And I cannot really explain what these graphs mean, but what you can see is that there are differences, right? Both in the soybean and in the corn plots, the 2 and the 4-year rotations differ in their microbial communities, both bacterial and fungal. So we had a lot of preliminary data, and what we did was we got together, myself and Matt Liebman, and with Larry Halverson, Gwen Beatty, and Phil Dixon, and Joe Hens, who's an economist, and we put together a proposal for NIFA, and we got funded. So this just started this year, and really the goal of that project is now to discover the mechanisms, what's really driving the suppression of SDS, and what can we learn from it and use it for growers. And so there's really 3 objectives in that project. The first one is we're going to collect a whole different variety of variables. So there's microbiome analysis, and more in-depth microbial community analysis, all the soil variables in nutrition, organic matter, everything you can think about, all the plant growth parameters and yield, and see what is associated with the healthy plots or the disease plots. The other thing we want to do is separate out the effects of oats and manure, because they are confounding factors in the trial, right? Because you can't really separate the oats from the manure. So we have field trials and greenhouse trials. We're looking specifically at those 2 factors. And we selected oats because they're known to have allelopathic effects. And also they're the common crop on the 3 and the 4 in the 3 and the 4 year rotations. So we hypothesized that was one of the driving factors here. And then finally, the economic profitability is looking at, we are applying seed treatment in all the rotation systems, and doing an analysis to see what is more profitable for growers. Is it to use a seed treatment in a 2-year rotation, or is it more profitable to not pay for the seed treatment and use a longer rotation? So long-term study will look at those factors. And with that, I just acknowledge my lab. There's a lot of people in my lab who help with a lot of this data. My collaborators for this project I mentioned, and then I work very closely in a lot of these things with Alison Robertson and Darren Mueller and Sylvia and Madan, Greg, and I'm sure many others that I didn't include here. So with this, I think I did okay on time. Time for questions. Any questions you have? Questions, yeah.
So in your new project, you said you're going to collect a lot more soil variables. In your preliminary analysis, did you look at soil types? In your plots, I guess, where in Iowa were your plots, and how did the soil vary?
Okay, good questions. The plots are at Marsden Farm in Boone, so they're kind of between the agronomy farm and the field station. Matt Lehman has all the plot maps, so there's several types of soils, they kind of wave in and out. These are very large plots. And so we have that characterized. There is, I think, one block that has a slightly different type of soil, but the rotation treatments are very consistent across the plot. So we think that at the soil type level, we don't think that's playing a role. But we're going to do a full analysis of everything. Yeah. Mm-hmm.
So when you do the SDS toxin screens,
Mm-hmm.
do you still get the interrenal browning that you do when the pathogen's there?
Yep. I don't remember if that picture maybe didn't show it very. Yeah, we can have very severe intervention. It won't defoliate. I mean, we manage the toxin. Yeah, but we get very typical symptoms. Yeah. Mm-hmm.
So what about the crop rotation that changes these? Because clearly there are family spores that can last longer.
Mm-hmm.
So what about oats or clover that changes the genes of the soil?
Right, that's exactly the question we're asking. So what is it about the rotation that's changing? The alfalfa and the clover, we know are hosts. We've done other studies looking at, and others have also shown this, that those legumes can get infected. So we don't think that the fact that there's clover or alfalfa roots in that system that is going to suppress a pathogen. If anything, it might build it up. So the oats might play a role. They don't get infected. And may have some compounds that are excreted into that environment, maybe killing or suppressing germination of the spores. There's definitely a difference in the pathogen load. Of course, we also have a gap, right? We have a 2-year rotation. We're only seeing corn and soybean every 2 years or every 3 years. So is it that there's just that space? So we're going to also, I didn't mention it here, but we're going to monitor the population changes over the different crops. So for each block, we're going to say, okay, what happens when you plant corn? Does it go up or down? What happens if you plant clover? Does it go down? And so we have, and the composts, composts have a lot of microbes. They have a lot of properties. Is it changing the porosity of the soil? Is it changing the ability of the roots to grow faster, more healthy, larger roots? Yeah.
So there is an interest in looking at soil microbiome in many places.
Mm-hmm.
So are you interested in doing something like that to see what changes?
Yeah. So objective 1, a big part of it and a big part of the funding goes for characterizing those microbial communities between the plots. And we already know there's some differences, but we want to know what groups of organisms are associated with one treatment or the other. Yeah, to see, and maybe we could start going in more depth in those and figure out if there's a biocontrol agent that's very active or any antibiotics that are being produced in the soil. Yeah, so from there we can go further. Yeah.
Let's thank Leonor one more time.